Assalamu alaikum. In this lesson, we are doing objective 16.2 part D, and we'll see how chi-squared test is used to test the significance of differences between observed and expected results. And the thing is, the formula for the chi-squared test will be provided to you in the examinations, right? But we'll see how it is used in genetic process, right? So let's go to the slide then. So chi-square is a type of statistical test through which we actually look for the relationship between different variables, right? So in genetics, we use genetic diagrams or genetic crosses to look at those relationships. So basically, what these tests assess is that whether the deviations from the expected results are significant or simply due to chance. Like, for example, while you're investigating something, you expect some results, you expect to get some results, right? But it is not, you cannot get 100% result basically, because definitely you are expecting something, but there might be a difference in your observed results, right? But after the experiment, if you look at your results, they might not be exactly the same as you were expecting, right? So this is what we assess in these chi-square tests. We see whether the deviations from the expected results are significant, to whether the result that we got, ठीक है, उसके अंदर जो हमारे पास values आ रही हैं, ठीक है, are there any deviations from the expected results? So are those deviations significant, ठीक है, or are they simply due to some chance? Right? So they determine the probability of results being due to chance. So chi-square test basically determines the probability of results being due to chance. This is what it actually gives us. When we look for genetics, expected results are actually like we can get them easily, right? But the observed results will be very different, right? And observed results different होने की major वजह ये है because like these are basically chance events, right? So if you talk about meiosis ke andar, like you have independent assortment can happen, crossing over can happen. So there are a lot of ways to this genetic variation can occur. Even fertilization, like random fertilization can happen as well, where X and Y carrying sperms have equal chance of fertilizing the egg, right? So all these actually chance events, they will definitely give you a different result as compared to what you were expecting, right? So this is what we expect in genetics, right? We expect these results to happen, right? So for chi-square test to begin, basically, or whenever we're conducting a statistical test, we always start by setting up a null hypothesis, right? So we always start by setting up a null hypothesis. So null hypothesis is actually used to determine, or you actually what you say, use a statement, right? Because it's a hypothesis. You use a statement, for example, like for instance, if you're saying that the observed results, they're not significantly different from the expected results, because this is what we expect. We say that this is our expectation, okay? And if we see that there is not much difference between the observed results, okay, experiment के बाद, when we're looking at the results, अगर उनमें और हमारे expected results में, if there is no such difference, there is no significant difference there, ठीक है? So ऐसे case में we would say कि yes, our hypothesis was correct, या null hypothesis was correct, ठीक है? तो हम null hypothesis को accept कर लेंगे, ठीक है? Because this is सबसे पहले बनाई जाती है null hypothesis, and we say your observed results are not significantly different from the expected results, right? So hypothesis के बाद, then you work through the statistical test, okay, and then you see whether the probability of your null hypothesis was correct or not, right? So in statistics again, or right now, because we'll focus on biological aspects basically. So the probability that we take as like cutoff जो हम लेते हैं 0.05 is the probability, ठीक है? Or you may mark it as 5%, right? So this is the probability. अब हम ये कहते हैं कि if the null hypothesis you're saying कि हमारी जो probability वो 5% है या उससे ज़्यादा है, ठीक है? If it is equal to, if it is greater than 5%, we will accept the null hypothesis, right? हमारी null hypothesis क्या है कि observed results are not significantly different from the expected results, right? So in that case, you are accepting the null hypothesis, right? It means if your value is 5% or more than 5%. However, if after the investigation, the observed results, you're getting the probability less than 0.05%, right? sorry, less than 5%, or the probability is 0.05, then you're saying okay, you are going to reject the null hypothesis, right? Because differences which are even smaller than 5%, ठीक है, तो उस case में there is a chance कि meiosis के अलावा something else has happened, ठीक है, there is something non-random that happened, right? Or maybe because like when we talk about like autosomal linkage, so those traits have been like inherited together, ठीक है. So meiosis के अलावा कोई और चीज हुई है, ठीक है, something that is non-random. meiosis में भी talk about कि random assortment हो रही है, like crossing over हो रही है, ठीक है, that random fertilization हो रही है, ठीक है. But अगर result 5% से less आ रहा है हमारा, in that case we are going to reject the null hypothesis, right? Because null hypothesis in this case it said that observed results are not significantly different. This is a null hypothesis, ठीक है. But अगर तो ये value 5% से कम आ रही है, it means there is definitely something significant, ठीक है. So we are going to reject the null hypothesis, right? So chi-squared test, ठीक है, it is written like this as well, x squared, right? So this is the term that we use, chi-squared test. All right then. So again, for clarification, if your null hypothesis is supported by the analysis, okay, you conducted the experiment and a null hypothesis is being supported, right? The assumption that mating is random, because this is what we assume, this is bound to happen, right? Mating is random, okay? Normal gene segregation, okay? Crossing over, independent assortment is bound to occur. Okay, we are assuming this should happen, this happens in meiosis, right? So this is the assumption in all the genetic crosses. We know okay, this happens, right? This is normal meiosis occurring and we would expect random segregation, random assortment, right? So if we are selecting or accepting the null hypothesis, or if a null hypothesis is being supported, okay, so this assumption, it means we are assuming these things have already happened, okay? If the null hypothesis is not supported, okay, by analysis, then the difference that you're looking at between the observed and the expected values, it means you're looking at a greater difference. Difference बहुत ज़्यादा है, ठीक है. Null hypothesis support होने का मतलब है there is not major difference between the expected results and the observed results, ठीक है. There is not the जो भी आपको difference नजर आ रहा है, it is not significant, या वो इतना छोटा difference है कि it can be neglected, ठीक है. But if you're looking at the null hypothesis which is not supported by analysis, it means that the difference that you're looking between the observed and the expected results is very different. Okay, the values are far apart. It means definitely something non-random must be occurring. Remember, हम लोग जब null hypothesis को accept कर रहे हैं, we are supporting it, we are assuming that these things are happening. Crossing over is happening, random assortment is happening, ठीक है, and this random mating is happening, ठीक है. We are actually assuming that. But if you're not getting such results, it means something non-random is happening. So possible explanation क्या हो सकती है? अच्छा ठीक है, non-random हो रहा है तो हम non-random है क्या चीज, कैसी क्या चीज हो रही है? It means genes are not randomly segregating, ठीक है, because either those are sex-linked, ठीक है, or they are linked on the same chromosome and inherited together. So these could be the other possibilities, right? So अगर हमारी null hypothesis is not being supported by analysis, the result you're getting is less than 0.05, ठीक है. ऐसे case में you're looking at maybe something that has been sex linked, or maybe the जो आपके जो links हैं, they are on the same chromosome, ठीक है, autosomal linkage है, and they're inherited together. So these are the two possibilities as well, right? So I hope you understand the difference between null hypothesis being supported. ठीक है, null hypothesis being supported means there is no significant difference between the expected and observed results, and this is what we were expecting. कि meiosis जब होगी तो these things are bound to happen, ठीक है. But if it is not being supported, it means something non-random has happened, and the difference between the expected and observed results is quite high, ठीक है. और इसकी possible explanations क्या हो सकती हैं कि maybe जो भी इसके अंदर genes हैं, they are sex linked, ठीक है, or there is some autosomal linkage going on there, right? All right then. So in this chi-squared test, what are we looking for? We are testing the difference between the observed results and the expected frequencies, right? So we are actually looking and using the symbol O for observed and capital E for expected frequencies of nominal data, right? And E stands for the predicted value on a theory, because this is what you're expecting. Like you predict something before in an experiment, you predict something that we'll get this much value, right? So जो हमारी predicted value है, that is, ठीक है, our expected value. So we will use capital E for that, and O is the actual value, the actual observation, the actual result that you get from the experiment, right? So these are the symbols that we'll be using. For example, if let's say you had 220 office springs, right, and from these parent plants, right? So we had, so this is heterozygous, right? So capital P stands for purple, small p stands for white, but definitely it's heterozygous, so purple color, right? And then we have two small p's are giving us white, right? So when they're mating, and like you draw up a net square, right? So you are getting 50-50%, like purple, heterozygous, and you're getting homozygous, recessive, white, right? So the phenotypic ratio that we're getting is 1:1 or 50%, right? So if you are having 220 office springs, right? 220 office springs were produced. So it means if the ratio is 50-50%, so there should be 110 office springs with purple flower, because 1:1 का ratio है, तो ये half हो जाएगा. ठीक है? So you should be having 110 with purple flower and 110 with white flower. ठीक है? This is what we are expecting. ठीक है? This is our expected value. We are saying if 220 office springs are produced, क्योंकि genetic cross के बाद हमारा ratio 1:1 का आ रहा है, so it means we should have 110 purple flower and 110 white flower. But when we get the actual result, ठीक है, actual result हमारे पास 124 plants का let's say आया, ठीक है, तो हमारे पास इस पूरे मतलब 220 के अंदर से basically 124 plants आपके पास purple आ गए और 96 आपके पास white आ गए, ठीक है. हम क्या expect कर रहे थे कि you should get 110 purple and you should get 110 white. But this is not the case. अब observed results में you're getting different values, right? So अब हम इसकी basis पे we'll see कि whether our prediction was wrong, ठीक है? Whether there's a huge difference between these results, ठीक है? और whether हमारे जो results आए हैं, whether they disprove the theory. So this is what we look for, right? In chi-square test. All right then. अच्छा, अब now we are using this chi-square test to compare the values, ठीक है? We had our expected results, ठीक है? And we have already had our observe results as well. ठीक है, हमारे पास दोनों results आ गए. अब हम इनको आपस में करें compare, right? So the result, and now what we do, we like draw a square, like अपने box बनाया, and then you know, okay, this is what you actually got, ठीक है, and this is what you were expecting. So what are we looking for? यहाँ पे the difference if you're looking at is 14, ठीक है, यहाँ पे like here the difference is also 14. ठीक है, तो मतलब expected में you were expecting more, you got less of white, but यहाँ पे हम expect कर रहे थे कि we will get lesser purple, but हमें ज़्यादा मिल गए, ठीक है. So the result will show us whether the difference between observed and expected is significant. Now if your difference is not significant, ठीक है, so अब उसके क्या reasons हो सकते हैं कि maybe the difference is so small that it can be neglected, ठीक है, or it means the result of the experiment does not disprove the theoretical prediction. तो अगर तो हमारी difference they are not significant, so it means we are not disproving the theoretical prediction because this is what we predicted. ठीक है? Okay, so this is how we do it. So we have the same box now, and we know we have this is our observed results and these are expected results. So सबसे पहला काम जो करना है that is state the null hypothesis. ठीक है? Null hypothesis is written as H dot या H naught. ठीक है? So your null hypothesis would be a statement. ठीक है? The difference between observed and expected values is insignificant. ठीक है? This is always our null hypothesis because we are assuming meiosis के अंदर ये तीनों चीजें होंगी, मतलब क्रॉसिंग ओवर होगी, इंडिपेंडेंट सेग्रीगेशन हो रही है, रैंडम असॉर्टमेंट हो रही है. वी आर एक्सपेक्टिंग दिस वुड हैपन. ठीक है, सो हमारी नल हाइपोथेसिस वुड बी दैट द डिफरेंस बिटवीन ऑब्जर्व्ड एंड एक्सपेक्टेड रिजल्ट्स इज इनसिग्निफिकेंट, राइट. So what we are looking for कि whether our null hypothesis is rejected or it is accepted. ठीक है, हम null hypothesis को accept उस सूरत में करते हैं when your observed and expected result is not significant because it is actually supporting our statement. हमारी statement यही है, the difference between observed and expected results is insignificant. ठीक है, and so if you're getting such results, you're going to accept the null hypothesis, right? But you're going to reject it if the difference between observed and expected values is insignificant. So it's significant. Significant, there is some major differences you're looking for. Okay, if अच्छा खासा huge difference है, that's something that you cannot neglect. उस केस में you're going to reject the null hypothesis. ठीक है, so ये हम दो चीजें इसके अंदर करते हैं, whether we are supporting, whether we are not supporting. ठीक है, फिर supporting के बाद we have to give reasonings as well. Okay, so first job क्या था? You state the null hypothesis. ठीक है, Second stage is you have to use this formula. So again, this formula will be given to you in the paper. So you calculate the chi-squared value, right? So here we were given the observed value. These are expected value. You use the formula, right? So for you do individually for all of those. For example, 124 minus 110 gives you 14. So you work out the square of 14. Okay, you divide by the expected value, right? And you get one value. Then you do the same for the white. You get another value. And finally, the formula states that it's summation of all these values. Summation means we add them up, right? So you add both these values, 1.782, 1.782, and you are getting this value. This is your calculated chi-squared value, right? This is a calculated value. All right. So this is a formula. It's summation of observed minus expected. You take the whole square, you divide this value by expected results. You do it individually for each category, okay? And then you add them up. This is how we calculate that. All right then. After that, once you're done with the calculation, and this was our calculated answer, third thing you have to do, that is you need to calculate the degree of freedom. Degree of freedom is something like, you know, you have number of categories. For example, in this case, purple category, white category. So हमारे पास number of categories, ठीक है, we denote the letter N. So we have the number of categories as 2. ठीक है, so the formula is for degrees of freedom, it's N minus 1. Right? So if it's N minus 1, ठीक है, so what you do, you had two categories. You had two categories, so 2 minus 1 gives you a 1. Okay, so this is a degree of freedom. And this is what we look at a table. Okay, so this is a table where we get the critical chi-squared value. So this table, okay, it has all the values here. We call this degree of freedom, right? And this has all the probabilities here at the top, right? And these values have the critical values here. Right? So we find the critical chi-squared value from the table. So the values that you're looking at here, these are known as the critical values, right? So again, just recall, we discussed that the percentage or the probability that we use, especially in the stats, and then definitely in biology, we go for 0.05 probability or we go for 5%, because it's the same. You're writing 5% as a fraction, as a decimal, in probability is 0.05. So we look for critical value at 0.05. Okay, so see, your value is coming as 3.5664, sorry, and this is the critical value that is here, 3.841. So your answer, your calculated value is less than this value. Your answer is 3.564. It is less than this value. It means it should fall along the left hand side. Okay, so if it is falling along the left hand side, it means, like, if this is 5%, okay, you're looking at the percentage is increasing in this direction. Okay, so it means it is definitely more than 10%. Your आपकी statement क्या थी? When you said your आपकी null hypothesis क्या थी कि there is no significant difference. ठीक है, and this is what you're looking for. बहुत ज़्यादा major difference नहीं है, 3.841. ठीक है, and your calculated value is 3.564. ठीक है, in this case, ठीक है, or especially if you're looking at this table, ठीक है, and your values are going towards the left. मतलब आपकी value, it is smaller than this value, 3.841 इधर है, and you go towards the left, you see values are getting lesser and lesser. ठीक है, in this case you're going to accept the null hypothesis. ठीक है, तो जब भी value 5% से ज़्यादा है, ठीक है, 5% से ज़्यादा है या 0.05 से ज़्यादा है, you're going to accept the null hypothesis, right? Had this value been more than this value, ठीक है, if it was going towards the right hand side, this is what you have to remember in biology especially, they are going towards the right, it means the values are smaller than 5%. ठीक है, यहाँ पे आप देखो तो it is 2.5%, it is 1%, ठीक है, it is even smaller than that. So if they're going towards the right here, in that case you're going to reject the null hypothesis. ठीक है, so just simply that something that you can use to remember कि कहाँ से हम accept कर रहे हैं, कहाँ हम reject कर रहे हैं. तो अगर वो right hand side पे आ रही है, because if you see here यहाँ पे values are getting greater and greater. ठीक है, so it means there is some non-random event that is happening. ठीक है, so जो भी आपका difference इतना major इसलिए आ रहा है, तो it means कि genes may have been sex linked, ठीक है, or there is some autosomal linkage taking place, ठीक है, मगर if you see something like this, ठीक है, values are smaller, ठीक है, and in that case you are going to accept the null hypothesis. इसका मतलब है yes, जो हम वही assume कर रहे थे कि meiosis के अंदर ये सारी चीजें होंगी, ठीक है, so those things have actually taken place. So in that case they are going to accept the null hypothesis. So if your calculated value is more than the critical chi-squared value. Remember, calculated value is the one जो हमने यहाँ पे टेबल पे सॉल्व की है, ठीक है. critical chi-squared value is the one जो हम यहाँ पे टेबल के अंदर देख रहे हैं, that we are looking at here. This is the critical chi-squared value. ठीक है. So हमने क्या किया, we worked out the degrees of freedom. We had two categories. ठीक है. So हमारे पास 2 minus 1, क्योंकि n minus 1 is the formula, we got 1. तो फिर हमने क्या किया, हमने critical अपने यहाँ पे degrees of freedom में 1 identify किया, ठीक है. 1 के अंदर then we look for the value, और हमें ये पता है कि we have to look for at 0.05. तो हमने ये देखा कि 1 पे the answer is coming at 3.841. This is your chi-squared value, critical chi-squared value. जो चीज आप टेबल में देख रहे हैं, this is critical chi-squared value, right? The one that you have calculated is your calculated chi-squared value. तो हम अपनी calculated chi-squared value को compare कर रहे हैं critical chi-squared value के अंदर, right? So if your calculated value, right, is, let's talk about this one because this is ये हमारा result है ना. हमारा calculated result was less than critical chi-squared value, right? It was less than critical chi-squared value. See, this was your calculated value. This is a critical value. Your calculated value is less. Okay. So in that case, your probability is greater than 0.05. Okay. Remember, 0.05 is 5%. Okay. Or इसके अलावा if it is more than 5%, so definitely if you look at this table again, okay, let me use the laser pointer here. See, so more than 5% is going towards this direction. Your answer is less. It is less than 3.841. It means the probability is more than 0.05 or it is more than 5%, right? Okay, so in that case, you're going to accept the null hypothesis. ठीक है. तो रिमेंबर टेबल पे अब गौर से देख लेना कि इन्होंने वैल्यूज किस तरह शो की हैं. ठीक है. इट्स नॉट ऑलवेज, ऐसा नहीं होता कि कभी ये भी होता है कि स्मॉलर वैल्यूज विल बी गिवन हियर एंड लार्जर वैल्यूज विल बी गिवन हियर. सो मेक श्योर यू लुक एट दिस टॉप पार्ट. ठीक है. इन दिस केस, ठीक है, हायर वैल्यूज आर ऑन दिस साइड, सो वी आर गोइंग टू एक्सेप्ट द नल हाइपोथेसिस. ठीक है. और इफ योर कैलकुलेटेड वैल्यू इज ग्रेटर देन द क्रिटिकल वैल्यू, इफ दिस वैल्यू वाज मोर देन दिस वैल्यू, तो इसका मतलब है your probability is less than 0.05, you're going to reject the null hypothesis. So have a look here again. ठीक है. If this value was greater, देखो यहाँ पे values greater होती जा रही हैं. So if our answer, our calculated value was more, ठीक है, तो it means probability जो है वो less than 0.05 है, and you're going to reject the null hypothesis. ठीक है. So we have already talked about that. ठीक है. अब हमारा answer, if you look at this answer, your calculated value was smaller. ठीक है. So our prediction, we said that there is no significant difference between the expected and observed result, and we got the same. ठीक है. So in this case, we are going to accept the null hypothesis. ठीक है. So जो smaller differences हैं इसके बीच में, it is simply due to chance. ठीक है. और due to chance की जब हम बात करते हैं तो we talk about the same: crossing over, independent assortment, ठीक है, random mating, ठीक है, and it could be sampling error as well. ठीक है. तो ये purely due to chance है. We are going to accept the null hypothesis. I hope it's clear. All right then, let's discuss this example is from the book. ठीक है. So generally जब हम बात कर रहे थे chi-squared की, तो questions आपको कैसे आ सकते हैं? You can get monohybrid cross करने के लिए आ सकता है. ठीक है. You can get something dihybrid cross के अंदर भी आ सकता है. ठीक है. और even codominance के questions भी आ सकते हैं. ठीक है. So for example, if you look at this here, so they are having two plants, ठीक है, which are heterozygous, right? So what they have done is they have taken a plant basically, और यहाँ पे dihybrid cross कर रहे हैं. So we are looking for flower color and the petal size. ठीक है. So ठीक है, दो traits हम लोग check कर रहे हैं यहाँ पे. So there for flower colors, allele capital Y, sorry, is for yellow, and small y is for white. और petal size है P, capital P is for larger petals, small p for smaller. ठीक है. So जब when two plants are crossed which are heterozygous of both the alleles, ठीक है, and if the alleles are not linked, remember, ठीक है, when they're not linked, it means chromosomes या जो genes है basically they're on different chromosomes. ठीक है. Then we expect to see offspring with this phenotype. So remember, this is what we are expecting in meiosis, right? Because we are assuming कि जब हम ये cross करा रहे हैं, we are assuming कि these जो genes हैं, ठीक है, they are not on the same chromosome, they are not inherited together. We are also assuming कि these are not sex linked. ठीक है. We are assuming that these are on two different chromosomes and they are not related to each other, they are not linked. ठीक है. तो हमने heterozygous, ठीक है, हमने ऐसे plants को cross किया which are heterozygous of both alleles. So remember, if these alleles are not linked, ठीक है, that is the genes are on different chromosomes, we would expect to get this offspring. ठीक है, this is our expected ratio. So we are assuming it's a dihybrid cross, or dihybrid cross, remember this is a ratio 9:3:3:1, right? ठीक है, so what they do, ठीक है, they like इन्होंने offspring जिसके जब produce हुए, so what do they get? ठीक है, there were 847 in total, ठीक है, and this is what they actually get. This is their result. ठीक है, observation ये इनके observed values है. These are the actual values that they got after the experiment. Yellow large इनको 489 मिले, yellow small 151, white large 159, white small 48. ठीक है, this is the actual result that they got. ठीक है, this was the ratio that they were expecting. ठीक है, अब how, ठीक है, they have actually, this is what they were expecting. So expecting results को how you can assess, simply you know कि this is a ratio. ठीक है, so yellow large was ratio 9. येलो स्मॉल का रेशियो 3 था, व्हाइट का 3 था, व्हाइट लार्ज और व्हाइट स्मॉल का 1 था. तो फ्रॉम दिस वैल्यू यू कैन एक्चुअली वर्क आउट द एक्सपेक्टेड वैल्यू. ठीक है, आपके पास येलो लार्ज इज 9 out of 16, क्योंकि अगर आप सम ऑफ रेशियो निकालो तो 9 + 3 + 3 + 1 gives you 16. So if we try it like 9 divided by 16, because we are looking for yellow large right now, so 9 divided by 16 times 847. 847 इसलिए कर रहे हैं because we had 847 offsprings, and if you work out this value, ठीक है, let me try that as well. Right, so you're getting 476.435. So we'll use the whole number here. Okay, so the same, then you'll do the same for yellow small. So you will go for 3 divided by 16 into 847. You get 159. Then again, since it's the same 3, you get the same value. Then you try for this 1 divided by 16 into 847, and you get 53, right? So हम अपनी expected values निकाल रहे हैं through this ratio. So अब again exam में questions ऐसे आएंगे, so you would be like required to find the expected results. So just look at the ratio, ठीक है? And like question में it is not especially necessary every time you'll get a dihybrid cross. Different type के आ सकते हैं. So use a particular ratio to find your answers. ठीक है? So these are your expected values. अब if you look at these values, this is your observed value, ठीक है? And we have our expected values as well. Then you see whether there is a significant difference between these values or there is not, right? So what we are going to do, now we are going to like have this table, okay? Again paper mein you will have the same table more or less, okay? So some boxes are filled, kuch empty hote hain, which we have to fill actually. So what they have done, they have actually given you all the observed values, okay? These are your observed results. So we saw that in the previous slide as well. These are your observed results. ठीक है, expected results अगर तो नहीं दिए हुए, ठीक है, so you know the categories, ठीक है, you must have done the genetic cross, क्योंकि पेपर में part 1 might be कि आपको genetic cross बनाना पड़ेगा, you will have to find the ratio. So in this case the ratio was 9:3:3:1. So if they have not given the expected values, आप उसे मालूम कर लेंगे using the same method. 9 divided by 16 divided by the total, multiply by the total, then you'll do the same for the 3 and the same for the 1. ठीक है, so you'll get the expected values. और अगर given है तो given है तो like that's fine. ठीक है, next what you're going to do, ठीक है, because your formula is summation observed minus expected. Let me erase that. ठीक है, it's observed minus expected whole squared divided by the expected. ठीक है, this will be your answer. So what we do, next step हम क्या करते हैं, observed minus expected. So difference between 489 and 476 is 13. Then 151 and 159 is -8. ठीक है, जैसा भी आ रहा है आपका negative, positive, जो भी difference आ रही है, you write it as it is. Then we know कि square of anything, चाहे वो negative number का हो, चाहे positive का हो, it gives you a positive answer. ठीक है, so you take the square of those values. ठीक है, square निकालने के बाद then you use this formula, square then divided by the expected value, and you get these answers. ठीक है, you do individually for each category. These are categories: yellow large one category, yellow small another category, white large another category, white small is the fourth one. Okay, so you add all these up because this is what your formula says, summation of all these values. आपने सबको add किया, and this is your calculated chi-squared value. Remember, this 1.23 is the calculated value. ये आपने observed और expected से calculate किया है. What you do next? अब आप अपनी calculated value को you look at this probability table. ठीक है, पेपर में, ठीक है, the values are sometimes given to you also. So this is the probability table. ठीक है, तो सबसे पहला काम क्या करना है, उसके बाद आपको you have to work out the degrees of freedom. ठीक है, यहाँ पे आपके पास कितनी थी, 4 categories थी, 1 2 3 4, and the formula is n minus 1. It means you get 3. ठीक है, अब यहाँ पर आप 3 पे, ठीक है, you will identify 0.5, 0.05. ठीक है, again I said के be careful, and then like see here percentages are increasing on this side and they're decreasing on this side. So फिर भी एक दफा have a look. ठीक है, so 0.05 में you look at 3. ठीक है, 3 पे 0.05 पे you're getting the value 7.82. This is your critical value from this table. This is your critical chi-squared value, 7.82. 1.23 is your calculated chi-squared value, and then you can see the difference. ठीक है, 1.23 is a smaller value. ठीक है, and then you look at this side, values are getting smaller in this direction. It means it is even greater than 10%, क्योंकि 10% पे 6.25 आ रहा है और आपकी वैल्यू तो 1.23 है. It means हमें तो far towards the left जाना पड़ेगा. ठीक है, so in this case it is going towards the left. ठीक है, it is more than 5%. In this case you're going to accept the null hypothesis. It means there is no significant difference between the expected and the observed results. ठीक है, no significant difference. I hope you understand now. So this is another example from the book. They have again taken 4 categories: purple stem cut leaves, purple stem potato leaves, green stem cut leaves, green stem potato leaves, right? So these are observed values. ठीक है, then ठीक है, sometimes you will be given an extra column for expected ratio. So we know expected ratio dihybrid process के लिए it's 9:3:3:1. ठीक है, then you will work out the expected number. If it is not given, so you will work it out, right? O minus E, then find the square. ठीक है, then use the formula O minus E whole square divided by E, and then you add all of them up, so you get the summation. In this case, the answer is 0.79. Again, the strategy is the same. You have this table. You find n minus 1. सबसे पहले degree of freedom ढूंढना है. n minus 1 आपके पास again 4 categories है. 1, 2, 3, 4. n minus 1 is 3. आपने दोबारा 3 देखा. You look at 0.5. 0.5 पे critical chi-squared value 7.82 है. Your answer is 0.79. Even smaller. बहुत ही छोटा है. ठीक है. It means definitely it's way more than maybe 30-40% से भी ज़्यादा का हुआ है. Then you're in this case you're going to accept the null hypothesis. It means there is no significant difference between your observed results and your expected results. I hope it makes sense now. And this is one more example. So here you see, again, they're saying there are 80 offspring. Okay, and these are the expected numbers using the same ratios. Again, for a dihybrid cross, 9:3:3:1. Okay, so they have worked out the expected numbers. This is what they're expecting: 45 gray long, 15 gray short, 15 white long, 5 white short. Okay. But these are the observed results that they are getting. Gray long 54, gray short 4, white long 4, white short 18. Okay, this is the observed result. Okay, after using this expected ratio, they have worked out the expected values. और अब इससे इसको minus किया है, so we are looking at these values: +9, -11, +13. ठीक है, then again using the same strategy, यहाँ पे जो आ रहा है आंसर का calculated chi-squared value is 51.8. 51.8. ठीक है, says again we have 4 categories, so our degree of freedom is 3, n minus 1. So 3 पे we know we have to always look at 0.05. Your answer is 7.82. It is the critical chi-squared value, but your calculated value is 51.8. So now you see values are increasing on this side. ठीक है, in this case it means we will have to reject the null hypothesis. Null hypothesis हम क्या कहते हैं? There is no significant difference between the expected and the observed results. But यहाँ तो बहुत significant difference है. It's quite a huge value. ठीक है, so in this case you are going to reject the null hypothesis. It means there is something non-random happening. ठीक है, जब हम यहाँ पे बात कर रहे थे तो we are expecting meiosis, we are assuming meiosis will happen. So random fertilization, random assortment, random segregation is happening. ठीक है, but यहाँ पे something non-random is happening. तो नॉन रैंडम के अंदर आपके पास क्या फिर एग्जांपल हो सकती है? मे बी दीज जीन्स आर सेक्स लिंक्ड. ठीक है, दे आर नॉट डेफिनेटली फाउंड ऑन मे बी 2 डिफरेंट क्रोमोसोम्स. ठीक है, उसके अलावा ऑटोसोमल लिंकेज का भी चांस है कि वो भी हो सकती है. ठीक है, और मे बी दे आर इनहेरिटेड टुगेदर बिकॉज यू आर गेटिंग अ बिगर नंबर. सो यहाँ पे यू आर गोइंग टू रिजेक्ट द नल हाइपोथेसिस. ठीक है, सो वी हैव लुक्ड एट वैल्यूज. वी वर गेटिंग ए स्मॉलर कैलकुलेटेड chi-squared value. ठीक है, we look also at this example where we are getting a value that is more than the this critical value. ठीक है, so जब more आ रही है you are going to reject the null hypothesis. ठीक है, अगर कम आ रही है you are going to accept the null hypothesis. All right, then let's try this question from the past paper then. So they're saying we have 2 pure breeding strains of snapdragon, which is a garden plant, and pure breeding, right? So it means homozygous dominant, homozygous recessive. Then one of the strains, because both of them are pure breeding, one had red flowers, other had white flowers. The 2 strains were then cross-yielding F1 generation and all the plants were pink. Okay? So the parental generation, one was red, one was white, but the offsprings are pink. Okay? So we are looking at codominance. Okay? But let's see. So F1 generation was then interbred to produce F2 plants and the following colors. This is the observed values. Okay? This is what they get after when they were interbred. So red, pink, and white. Okay? These are observed values. 62 red, 131 pink, 67 white. So this is the hypothesis. Okay? This was hypothesis that was produced, and this said flower color is controlled by a single gene with 2 codominant alleles, right? Because this is what we have like learned in codominance. Okay? So both of them are dominant. Okay? So in this case, they are saying the flower color is controlled by a single gene. Okay? But alleles, both of them are codominant. So that's the reason we are getting a pink offspring as well. Let's look at the questions now. So first up, complete the genetic diagram to explain this cross. So again, in every question, you will not be given the ratio. You will have to find it first. ठीक है, and as I said earlier, every question will not be of dihybrid cross. आपको different questions आ सकते हैं. Monohybrid cross का भी आ सकता है. Dihybrid की we have just discussed the examples, and this question is of codominance. Let's see. ठीक है, so they say use the following symbols to represent the alleles. So ठीक है, C small r is for red, C W is for white. So this is the parental phenotypes. ठीक है, so remember the first up they said 2 pure breeding strains of snapdragon were obtained. One had red and one was white. So right now, first जो हम काम कर रहे हैं यहाँ पे, this is for the parental generation. ठीक है, and it's pure breeding. ठीक है, both of them are pure breed. Right, so parental genotypes should be for red flowers CR, CR, and for white flowers it should be CR, W, CR, W. Right? So these are the genotypes. Sure. When you look at the gametes, so gametes तो बनेंगे भी तो definitely CR, ठीक है, दोनों ही same है, CW, or you can write them like this and also, ठीक है, आपके पास ये gametes बन रहे हैं, ठीक है. Okay, so what will be the genotype? There's a space you can use for your genetic cross, right? So if we make a genetic cross, so CR, CR, then CW, CW, and we are expecting all of them to be heterozygous. Right? And we are looking at codominance, right? So the genotypes would be all of them are CR, CW, right? All of them are. And if you look at the phenotypes, all of them should be pink. So all pink. Right? So we have all of these heterozygous, all pink, and gametes. Okay? If you look at the gametes, so you are CR. Okay? ठीक है, CR, CW, ठीक है, क्योंकि हम आपस में इनका क्रॉस करा रहे हैं, ठीक है, so CR, CW. These are the gametes that we are getting now. Okay, let's look at the next one. So this was your F1 generation, ठीक है, F1 all heterozygous, all pink, and these are the gametes. ठीक है, now we are crossing these two, ठीक है, अब इनका क्रॉस करवा रहे हैं. So you have CR, CW, CR, CW, and if we do the crossing now, so we have CR, 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 CW, we have CW, CW, and we have CW, CW. So what are we looking at here? CR. Oh, my bad. Yes, I have to correct. This should be CRCW. Yeah, it's correct now. Okay, so your genotypes are, let's write them separately here. So the genotypes that we are getting CRCR, we are getting CRCW, okay, again CRCW, and we are getting CW. So if you look at the phenotypes, these will be red, right? CRCW, these two are pink, right? And here, white, right? So if you look at the expected phenotypical ratio, so we had one red, we had one white, but two of these are pink. So your phenotypical ratio is 1:2:1, right? So again, remember these are our expected ratios. This is what we're expecting. So this whole working for 6 marks. Okay, let's tally it from the answer key also. So we see how the marks have been distributed. So have a look at the marking key. So first we had to write the parental genotypes because initially this said that it's pure breed. Okay, so we had red and we had white. Okay, these were the gametes. And for writing genotypes and phenotypes, or both of these, okay, 1 mark for that. Okay, then after genetic cross, we get all of them pink. Okay, then you go for the F2 generation. Okay, they were all heterozygous. Okay, so in corpus में interbred कराया, so these are the gametes. Okay, and this is your F2 genotype. You're getting red, pink, and white. 1:2:1. This is the ratio that you're getting. Okay, so these were 6 marks. Let's look at the next part of the question. All right. Now they're saying a chi-squared test is carried out on the experimental data to determine whether the hypothesis is supported. Remember, hypothesis हमारी जो थी, it was already given to us. And it said that flower color is controlled by a single gene with 2 codominant alleles. So now we have to use a chi-squared test. Okay, we'll use the formula and then we'll see okay, whether we are accepting this or this hypothesis or we are supporting it. Okay, so let's look at the data now. All right then. So some parts are already done for us. So they are telling us okay, from the red flowers, we get 62, 131 were pink, 67 were white. Okay, so these are the observed results. Okay, expected numbers you have to work out. कैसे work out करेंगे? Remember, we had this ratio. We worked out the expected ratio, expected phenotypical ratio 1:2:1, and we had 3 categories. Okay, अब using the same 1:2:1. Okay, and you also know that we have total 260 flowers there, right? So use the same ratio to work out the expected value. So let's do it here. Since sum of ratio निकालेंगे, तो you're getting a 4. Okay, so I'm doing for red right now. So 1 divided by 4 divided by 260. Okay, let's see what we get. So 260 divided by 4, you get a 65. So expected number is a 65. Okay, let's do that for pink because the ratio is 2. So we should be getting a 130. Okay, and here it is the same 1 divided by 4 into 260 because the ratio is there. So you're getting a 65. So now even if you try to add them up, they'll add to 260. So this is how we worked out the expected numbers using this expected ratio, right? All right, then इसको भी tally कर लेते. So 65, 130, 65, and we got the same values, 3 marks. Okay, then see, the formula is already given to you. The chi-squared statistic is calculated by the following formula. Okay, and then they're saying calculate the value of chi-square from the above data, and then you have to show your working as well. Okay, So okay, the first step, ठीक है, आप यहाँ पे भी बेशक extra column बना लें, ठीक है. So we know कि we have to जो पहला काम करना है, that is O minus E. So let's work out the difference. It's O minus E, observed minus expected. So in this case, you're getting a -3. ठीक है. Here you're getting a 1 plus 1. Here you're getting a 2. ठीक है. So ठीक है, next step में क्या करना है आपको? You have to square these values. ठीक है, because we have O minus E whole squared. ठीक है, so -3 का square भी करेंगे, we are getting a 9. Square of 1 is a 1, square of 2 is a 2. ठीक है, और next step में क्या करना है? This value divided by E, divided by the expected value. So it's 1, 2, 3 here. ठीक है, so we know according to the formula O minus E whole square divided by E. So let's do that. ठीक है, you'll definitely get negative values. So let me work that out. This is 9 and your expected value was a 65. Okay, so let's see, 9 divided by 65. We get 0.138. तो चलो हम 3 decimal places तक use कर लेते हैं. It's 0.138. All right, for here, it's again O minus E whole squared divided by the expected value is 130. So let's see, 1 divided by 130, you get 0.00769. So we are getting 0.00. Okay, it's 769. So we can round this as well. It's 769. So 760. And last bit that you have is ticket 2 and this should be a 4. Let me correct that. 2 का square 4 होता है. Okay, so this should be a 4 and 4 divided. So yeah, 4 divided by 65 gives us 0.0615. It can be rounded to 0.062. So let's just add all these three. So we have 0.138 plus 0.0077 plus 0.0615. Let's all these up. 138 plus 0.0077 plus 0.016 by 0.0615, and we are getting 0.2072, or we could just round it to 0.207. So let's tell you the answer key, and this is the range of answer, 0.206 till 0.208. Okay, we got 0.207. All right then. Okay, then they're saying the critical value of chi-squared for this type of investigation with 2 degrees of freedom. 2 degrees of freedom, remember we had हमारे पास 3 colors, red, pink and white. So you have 3 categories. Okay, so n-1 degree of freedom is 2. Okay, so they are saying the critical value that you would be looking at the table. Okay, they've already given you this value 5.991. Okay, or again ये value हमें 0.05 के against देखते हैं. Okay, so this is 5.991 and your answer 0.207, or rather this is your calculated chi-squared value. Right, this is your calculated chi-squared value and this is the critical chi-squared value that we see in the table there, right? So if you compare your calculated value with this value, it is quite small, right? It means we are going to accept the null hypothesis. हमारे पास smaller values थी towards the left hand side, right? If I could show you the chart again, so see 0.05 is here and we have a smaller values. They said कि degree of freedom is 2 और उसके against 5.991 है, तो ये 5.99 है हमारे पास, ठीक है. Your value is quite smaller, ठीक है. So it means like your value is like 0.05 से definitely it's more than 5%, ठीक है. It is more than 5%. So now they're saying explain whether your answer to B, this are your calculated answer, it supports the hypothesis or not, ठीक है. So remember अगर हमारा answer 0.05 या 5% से ज़्यादा है, ठीक है, we accept the null hypothesis. So अभी ये 1 mark question and they are saying whether it supports. So first you will write yes you support or you accept or whether you reject. It totally depends upon your answer. Okay, in this case we are supporting, but then you have to give the reason as well. Why are you supporting it? Okay, 1 mark से इस बात का नहीं मिलेगा कि you're supporting it. Okay, so reason would be, okay, yes, our value, our calculated chi-squared value is less than 5.991. Okay, because this value is lower than the critical value. You are going to write this reason. Okay, you will not only write we support it. You have to give the reason as well. Okay, so you're saying that the figure, the answer that we got is less than the critical value. Okay. So I hope you have understood how you're going to solve these questions. Thank you.